Right, just a quick video to help with any potential questions with this. Now, these are all questions that can be found in the notes, so there's not really a whole lot to say about these, but as for these, it's good to just kind of be clear what we're looking for. So simulate DNA replication means just show what DNA letters pair with these. So T pairs with A, G pairs with C, A pairs with T, T pairs with A. So you would do the same here, like for example, C pairs with G, T pairs with A pairs with T, etc. You'd write all 6 letters here and then do the same for these. And then the same thing is going to occur with these other ones. Except, of course, make sure you do what it's asked for, transcribing the DNA into RNA, mRNA, which means write the RNA letter that pairs. So that means in RNA, C pairs with G, U pairs with A pairs with T, and so on for all 6 letters. So there should be 6 letters in your version of this, not just 3. All right, moving on. tRNAs are complementary to mRNAs. So if you have an mRNA sequence, then it's one RNA pairing with another RNA. So what that means is that's why it's A pairs with U pairs with A, C pairs with G. So you G, C, A, etc., etc., etc. There should be 6 here, not 3. This is just my showing the idea, and then do that for all the rest of these too. Translating mRNA into amino acids. You've got to look these up on the chart. GAG, you start with this one first, then this one, then this one. So if it's GAG, the reason why we said this one is because we did G, and then A, and G and A come together in this box, and then which of these 4 do you use? This G tells you use the bottom one. So where all 3 come together, it's this one. That's why you put GLU right there. So that's glutamine, and then serine is because UCA, U is the first one, C is the second one, so U and C point to this box, and the way you know which one to pick out of this box is to note that the last letter is A, so you look up A, SER, serine. All right, that's why that's that. So you follow this pattern in all of these. So GUG, you look that up. Can you tell what it would be? G is the first one, U is the second one, so it's somewhere in here, and then G is the last one, so it's more specifically this one right here, valine. So VA, oops, I just wrote that in the wrong one. VAL, and then you'll put a little dash in the next one, CCU, so look up C is the first one, C is the second one, so it's somewhere in here. U is the last one, you choose proline. It's not necessarily write out the entire name, but the abbreviation is fine, just like this. And you do the same thing for all the rest. Now here, mRNAs are complementary to the following tRNAs. So in other words, you're just taking writing the RNA letter that's complementary to the tRNA letters given. So U pairs with A, G pairs with C, A pairs with U, etc. Likewise, you'll put a G, and then a G, and then a C, and then a C, and then a U, and then an A. That's what you would write here, and you'd follow that same pattern for all the rest of these. On the back, you will need this for part of this. You do all of these. Now, you'll notice you can't really go from top to bottom because like there's multiple serine, for example. There's multiple codons that can produce a serine. So instead, you start here, and then following these directions, do the mRNA first, which means do this, and then the amino acid, which means and then this, and then the anticodon last, which is this. So first, second, the amino acid gets written twice, once here, and then third, you do this as an example. You do this one first, and then you do these next, and then this one last, because U pairs with A, G pairs with C, G pairs with C. You look the mRNA up on the chart, U, G, so U and G come together here. The third letter is G, so we look it up right here, TRP. I then would have written TRP here and here, and then because U pairs with A, the anticodon, A pairs with U, G pairs with C, G pairs with C, I would have written these in last. Let's do the example here. C pairs with G. A pairs with U, T pairs with A, GUA, I look it up on the chart. G, U comes together here. A is this one specifically, it's valine. VAL, VAL. Lastly, I write with the letters that pair. C pairs with G, A pairs with U, U pairs with A. And you do the same for all the rest of them. Here, you do a similar thing, except the only difference is you'll notice the mRNA is present. So you can look the mRNA up, look up what GGG is, and put that amino acid here, because this is the mRNA chart and this is mRNA. You'll fill in the DNA part that goes with it, so you would write CCC for the DNA. But you look up GGG, yep, should be that one. You'll put that here and here, and then you'll of course fill in the RNA letters here that match, etc., etc. Here you could do the same thing. You could fill in the mRNAs that pair with this. So you fill in the DNAs that pair with the RNA letters here. And then you can look up whatever letters you put here in the mRNA, you can look up on this chart, because this chart is only for mRNA, not tRNA. If you look up GAU, you're going to get the wrong thing. That's why, for example, here, GAU is for this one. So by chance these are the same, but that's only by chance. G, A, so G and A come together here. And then U is the last letter, so U is ASP. That's why I put asparagine here, or aspartame. And so that's why you would put this one here. It's because that's what this codes for this. Make sure that this also gets written up here. And then you're going to follow that example for all the rest of these. All right, any questions? Let your teacher know. Any questions, check with your partner. And then if you're both confused, let the teacher know. There you have it.